Thank you, Carol, and I'd like to thank the organizing committee for the kind invitation to speak here. I've spoken here once before, and it's always both an honor, a pleasure, and also a little bit intimidating to speak about immunology in front of a group like this. And I think of sarcoidosis as such a fun immunologic disease that I can't help but keep going back to that. It's a little like bringing coals to Newcastle, but in any event, we'll talk a little bit about where we are today with biologics and sarcoidosis. By way of disclosures, I'm involved in some trials and also have some research support, and I would point out that there are no FDA-approved medications for sarcoidosis. Interest in sarcoidosis therapies has been ascending, and that's in part driven by recognition that mortality rates are increasing, both in the US and abroad, specifically in Britain that's been looked at, and also the morbidity of the disease is increasingly recognized. Despite the ascending interest in sarcoidosis on both an academic, clinical, and a pharmaceutical level, there's been very little done in the arena of sarcoidosis therapeutics. And I just have some rough estimates here that I pulled in a very non-scientific way using some prevalence data and a search of PubMed, pointing out roughly that sarcoidosis has a prevalence similar to many other diseases in which there are many more randomized controlled trials in humans and many more FDA-approved medications. So in sarcoidosis, we actually have 0 FDA-approved medications and very few randomized controlled trials. And so when we have at least one randomized controlled trial, it's big news to us. And so we talk about trials one at a time instead of in aggregate like you guys typically do. And so with that in mind, this is the outline for my talk today. I'm going to talk a little bit about the experience of over a decade now using Infliximab and other traditional anti-TNF agents in sarcoidosis. And then I'll give you some unpublished data that have been publicly presented before on a recent trial in pulmonary and cutaneous sarcoidosis of golimumab and Ustekinumab. Now, sarcoidosis was first described about 150 years ago by Jonathan Hutchinson. This guy's interesting. He was a surgeon, a dermatologist, an ophthalmologist, and a pathologist. And he published over 1,200 articles. He was the first to really recognize sarcoidosis in the modern era, and he termed it Mortimer's malady. And this was a very phenomenologic description, but one of the things that he pointed out was that it had similarities to both tuberculosis and lupus. And I hope by the end of the talk I'm able to impress on you that that's probably still our current pathophysiologic understanding of the disease. The term sarcoid was coined by Cesar Beck about 20 years later, but despite Hutchinson's description of Mortimer's malady as a very phenomenologic disease, we have not really made a tremendous amount of headway in understanding the pathophysiology. So much of what we think about with sarcoidosis is still very descriptive and empiric. Here is a schema, and this is, I hope, the most complex one that I have today, showing a hypothesis, probably the most update and interesting hypothesis that I thought I would share with you about the pathophysiology of sarcoidosis. And I thought this was interesting because of the renewed focus and research in the role of the innate immune response in driving immune-mediated inflammatory diseases. And the latest hypothesis in sarcoidosis is bringing this back in through this pathway of hyperproduction of serum amyloid A and also some kind of antigenic trigger. In this author's schema, this is MCAT-G, mycobacterial catalase. But that these things combine together to drive the cell-mediated immune response that's typical of sarcoidosis, the granuloma, where perhaps there's poor resolution in part due to the amyloid A protein. And then that may in part explain why some patients who can resolve this amyloid protein aggregate are able to go on to resolve their disease, whereas others have persistent disease that continues to drive the immune response. The interesting thing about this hypothesis is that you do not see amyloid A deposition in other granulomatous diseases, including Crohn's disease, Wegener's granulomatosis, if you take that as a granulomatous disease, or even chronic beryllium disease, which I used to think was very similar to sarcoidosis, but with just a known antigen. So stay tuned to this story, because this may give us some specific therapy. Here's the way that we've thought about sarcoidosis pathophysiology traditionally. This is a cell-mediated response. There's your typical usual culprits there on the left side of the schema. There's some kind of poorly degraded antigen. And then we've thought of TNF as a big central player that's necessary to drive and perpetuate the formation of the granuloma. And for that reason, TNF antagonists were looked at very early on as a very important target for the maintenance or for the therapy of sarcoidosis. And of course, as you know, TNF has a lot of roles that you could hypothesize would be important in sarcoidosis. It's important for generation of reactive oxygen intermediates that are important in sarcoidosis, or cellular recruitment, and also there's a feedback loop with interferon gamma. And I'll show you some data at the end of this talk to suggest that interferon gamma, as you probably already know, is very central in the pathogenesis of sarcoidosis. There were nice in vitro data suggesting that TNF would be a good target for sarcoidosis. Here's the reduction of interferon gamma after stimulation of monocytes with MTB. And you can see that all of the three oldest agents all have some dose-related reduction in interferon gamma production. And so this led to the first large randomized controlled trial that was ever conducted in sarcoidosis that was published just 7 years ago now and involved 138 patients. And after this big effort, and a positive trial in fact, the collective response was, so what? I don't think anybody was excited by Infliximab in sarcoidosis. Nonetheless, we've adopted it. And in fact, we've adopted a range of TNF antagonists, and these are the ones that we currently use most commonly today. In my clinic, these are the ones that I focus on primarily. Some of these other ones may be useful. I think the story on rituximab is way too early to understand what it's going to do. And I would point out here, similar to Crohn's disease, that there's little role for etanercept in the treatment of sarcoidosis. It does not seem to be a very effective granuloma busting agent. So here's the thing I told you that everybody said so what to. This is the response in percent forced vital capacity after treatment with Infliximab in chronic pulmonary sarcoidosis. And if you look at the y-axis, you might say, wow, 2%, so what? Not a very big change. However, if you start to look at some of the patients with more severe disease, which is this more severe strata in the Bothman study, or a similar group which was studied in a smaller study, an open-label prospective study, a double-blind study by Milt Rossman, but very small, you start to see changes in the order of 5%. Now, in the IPF world, we would think a 5% change in the forced vital capacity is not a very effective change. But if you look at a change of 5% in somebody who has chronic, drug-requiring, multiple therapy-requiring disease who has progressed over time, this is actually, I would put to you, about as good as you could expect. And I'll show you some data in a minute to suggest that this is just about as good as what you get from steroids, which I think we all recognize are effective therapy for sarcoidosis. So one of the messages I wanted to communicate to you today is that the perception of Infliximab and other TNF antagonists perhaps in the therapy of sarcoidosis as not being effective for pulmonary sarcoidosis is probably more driven by the choice of endpoints in the trials than it is by the true effectiveness. Here are those data I told you about. This is the effect of steroids in our population in patients treated with prednisone for a few weeks, looking at the FVC and the DLCO. And you can see relatively modest effects on both the FVC and DLCO. The important thing about this is that all of those patients who had improvements of at least 5% in one or the other of these pulmonary function tests also had improvements in their quality of life and their dyspnea scores. And so, in fact, we're starting to think that 5% is a more reasonable target for trials and therapeutic trials even in the clinic when you're treating patients with chronic pulmonary inflammatory diseases. If you look at extrapulmonary sarcoidosis, we run into many of the same problems that some of the more complex rheumatologic diseases have, and that is that there are various manifestations and it's hard to collect them in one basket. Here's a metric that Mark Judson came up with called the E-post, and I have no idea what the word E-post stands for. I just know that it means, did the patient overall get better? But if you look at the E-post score in extrapulmonary disease, you can see fairly nice effects in terms of improvements in extrapulmonary disease, and that is certainly what we notice clinically when we treat patients with Infliximab. Now, what about stratifying patients? I know that that's something we sometimes try to do. Does CRP predict a response? Does angiotensin-converting enzyme level help you? And the second take-home message I want to get to you today is that there are probably no serologic tests that help you very much. Everybody likes to measure the ACE level, but here's one study from the Netherlands that looked at the ACE level in patients with and without respiratory function impairment and who required treatment or no treatment, and there's no difference in ACE level. ACE level, likewise, is not predictive of the course of the disease. So I think that if you want to measure ACE level, SED rate, CRP, you will probably miss some patients who can respond and also overcall some patients who may not respond to therapy. I tend to move away from measuring serologies. What about using a chest X-ray to guess who's going to respond to therapy? Here are data from our center again, looking at the response in vital capacity in chronic sarcoidosis, depending on whether or not you have a chest X-ray that would be predicted not to respond. Those are people with fibrosis on their chest radiograph. And you can see, I guess this is kind of a difficult slide to see, but you can see the change without a fibrotic chest X-ray, which is this middle bar, is, with the fibrotic chest X-ray is actually more robust than the ones who do not have a fibrotic chest X-ray. So I do not think you can use radiography alone to determine, at least chest radiography, to determine who will or will not respond to therapy. Maybe PET scan can do better, and that's an area of active research, but of course that's very expensive, and it'll be a while, I think, before we know that answer. So at the end of the day, we come up with sort of algorithms like this. Despite the lack of excitement about the use of Infliximab because of that trial and the relatively modest response, it still has a home in the algorithms that we've developed in the sarcoidosis community because of what we see in the clinic. And so we tend to use it typically as a third-line agent. There are some situations where I have started moving it into a second-line agent or even up front. And I think that CNS sarcoidosis that's severe and lupus pernio are the two of those where I tend to move very early on to TNF antagonists, simply because of the severity of the disease in the first case and the difficulty of treating it with any other agent in the second case. So here are the main points that we have so far. Number one, most of how we manage TNF antagonists in sarcoidosis is based on extrapolation. So for example, should we use methotrexate or leflunomide along with our TNF antagonists? Most of us do that, although there are very little data about the effectiveness of that, either in terms of the drug's effect or in terms of preventing anti-drug antibodies. There are some caveats to that, however. For example, there's a black box warning about using the TNF antagonists in severe cardiomyopathy based on some early cardiac failure trials and some case reports. Nonetheless, in cardiac sarcoidosis that's refractory to other agents, we have had some successes using TNF antagonists, and I think that I still consider that in cases where I can't control it with anything else. There's no comparative controlled trials for this. I guess this is probably a ubiquitous problem across all immune-mediated inflammatory diseases. However, anecdotally, Infliximab seems to work the best. This may have to do with PK/PD issues. This may have to do with the speed of response and our lack of patience. Nonetheless, this is the one that we tend to use in severe disease because we see the best responses to it. And finally, there are some important differences in response between organs, and I'll just show you two pieces of data about that right now. One of those is lupus pernio. This is a series that will probably not be repeated anytime soon. This is a series of 54 patients with lupus pernio who were treated in South Carolina with various agents, and on the x-axis they have, I don't know why they use this abbreviation, agent, for cytotoxic like methotrexate or azathioprine, corticosteroids alone, corticosteroids with another agent, or Infliximab. And you can see that in monotherapy with an agent like methotrexate, there was very, very poor responses, whereas Infliximab tended to be much more effective. Corticosteroids are fairly effective for chronic cutaneous sarcoidosis, especially lupus pernio, but generally only in quite high doses that are very difficult for patients to tolerate over the long term. So if you see a patient come in the door with lupus pernio or very severe cutaneous disease, I would think early on about whether or not a TNF antagonist is appropriate for that patient. Here are some data that are just retrospective from our center. looking at the response in various organs. And the point here is that the pulmonary disease is actually a relatively less easy organ to see responses in as compared to CNS disease or skin disease, in whom you often see quite dramatic responses. We had a small series a few years ago looking at patients who were failing cyclophosphamide for severe CNS sarcoidosis, but who yet had dramatic responses to Infliximab. And for that reason, we have moved Infliximab early on into our protocol for severe central nervous system disease, sarcoidosis. All right, so let's move on to the next topic here, and I'll show you this trial. This is not in any of your slide set. And this is a trial looking at another TNF antagonist, golimumab, and a P40 antibody, Ustekinumab. And you're well enough aware about the role of IL-12 and IL-23 in inducing the immune response and in driving the interferon response, which ultimately results in granuloma formation in sarcoidosis. And there are plenty of data showing that IL-12 levels are associated with the prognosis and activity of the disease, and that IL-12 drops as the patient resolves their disease. And so there was some excitement about blocking this pathway as a mechanism for treating sarcoidosis, especially in patients who may not tolerate or who had failed TNF antagonists. Here are some data looking, just as proof of concept, looking at TNF and IL-12 levels in the skin. And if you look in normal skin of sarcoid patients, there's relatively smaller levels of TNF, similar to healthy controls, as compared to lesional skin. And so again, this is proof of concept data suggesting that these agents may be effective for sarcoidosis. The other thing that happened with this trial is there was an attempt to enrich the population. And that's always the response when your therapy doesn't work, right? So we did something wrong with the endpoint, or we did something wrong with the population. Let's find a better population in which to really show our drug is effective. And so what they did is they took the initial Infliximab trial, which had 138 subjects, and then they started to look at which groups were the most responsive. And they found that those with lower vital capacities and lower 6-minute walk distance started to see bigger changes in FVC. If you had more chronic disease, that enriched the population even further, bigger changes in FVC. And then if you had dyspnea, that even enriched the population slightly more. So this became the entry criteria for the trial that I'm going to show you. Chronic disease, substantial impairment of pulmonary physiology, and some symptoms as well. And so the thought was that this would be the one easiest to show response in. Now, as you know, P40 is a subcomponent of both IL-23 and IL-12, and Ustekinumab is a molecule that blocks P40, preventing signaling into the T cell or the NK cell. Here's the design of the study. This is a 16-week active treatment phase. A 28-week active treatment phase, but the steroids were held stable for 16 weeks, and then there was a steroid tapering phase. And I'm not going to show you any data about the steroid taper, except to tell you that there were trends, not statistically significant, but very nice, close to statistically significant trends of reduction in steroid dose, at least in the golimumab arm. And I guess that presages the results of this study. Here's the way it was designed. Some patients had lung disease alone, some patients had skin disease alone, and some patients had both skin or lung disease. You can see there were only about 17 patients, and I think a number of the ones that were enrolled here, in fact, fit into that stratum. And so these were analyzed separately, but the primary target was the lung disease. And here are the data, and this was a bit of a disappointment. You can see that golimumab, actually both of these look, at least in this analysis, to fail as compared to placebo. Certainly Ustekinumab looked even worse than golimumab in terms of vital capacity. If you look at responder analysis, taking that 5% threshold that I told you about, again, no difference between the groups. And then what about skin disease? We certainly saw some patients in the trial who had some substantial improvements in their skin using something that was developed for sarcoidosis called Skin Physician's Global Assessment. But if you look at the analysis, you can see that there just was not maybe perhaps enough to see the effect of golimumab. and the hypothesis that blocking P40 would be useful in sarcoidosis, at least using this particular strategy and this kind of endpoint, was not proven. And so the momentum for that sort of approach has now abated. In the last few minutes, I want to talk about one more thing, and that is a little bit about what we've learned about granulomas as a result of our experience with TNF antagonists in the treatment of immune-mediated inflammatory diseases. And although this is probably not going to be very good for these guys, we've tried to learn a little bit from the notion that everything is not always as you think. And the story is going back to the idea that TNF is so important in the maintenance and formation of granulomas. And you guys have probably seen data similar to this or this particular data before, but this is just a TNF knockout mouse showing that on the left, under an infectious disease model, granulomas form very nicely. If you knock it out, there's very little granuloma formation. There was an interesting report by a guy called Claude Milman in Denmark who gave a single dose of Infliximab to a patient who was waiting on the lung transplant list. And the patient got a transplant much faster than they expected to, so they got an opportunity to look at the allograft the explant right after the transplant. This is a person who had FDG positivity in their lung during the transplant workup, got a dose of Infliximab, had their lung come out. There were essentially almost no granulomas left in the lung. So we know that TNF antagonism can be quite effective at reducing the formation of granulomas. However, it seems that in some patients there's an escape, and in some patients there's relative refractoriness. And why is that? And I think part of the answer to that story might be in this observation about paradoxical granuloma formation in patients on TNF antagonists. And you guys have heard this story before, but this is a patient that I ran into last year, and I just put it up here for illustrative purposes. This is a woman with IBD on adalimumab who developed fairly bulkier adenopathy than we would typically see in IBD and fairly better formed granulomas in the mediastinum than you might blame on IBD. And of course, this is a phenomenon that's been trickling into the literature over the last 11 years, and I think that we all recognize that this can actually happen. If you look at this, it's a relatively unusual phenomenon. It's been reported most often with etanercept. Perhaps that's because etanercept tends to perhaps be a little bit better at inducing some type 1 interferon signatures than the antibody therapies do. It typically is described as affecting the lungs, but may involve other organs. We've had at least 2 patients that I've seen with uveitis as a result. And in fact, the uveitis has been relatively more bothersome than the lung disease, and in one case led to blindness after use of etanercept. Most of the patients have had withdrawal. And if I can make a plea to you, please don't call this sarcoidosis. Most of my life is running around trying to erase diagnosis sarcoidosis from the charts of patients who have it, and I just wouldn't call this sarcoidosis. But anyway, it's some kind of granulomatous reaction. So why does it happen? And you've seen this schema before. But I was struck looking in through some of our explants that there are a large number of patients with active granulomas still in the lungs despite therapy. Now, it could be that the therapy simply is not effective enough. It could be that there's a dosing issue. There could be a number of reasons why. But it's interesting, even in patients who have had some response and no evidence of antibody formation, that there are active granulomas. And it raises the question about whether TNF in chronic treatment is really as important as we thought it was. And it may be that as you get some of these shifts that were described yesterday away from TNF to maybe more of an interferon signature, that there's an alternate mechanism perpetuating granulomatous inflammation. And certainly that would accord with the sorts of case reports that you hear about where people treated with interferon alpha and developed granulomatous disease that resembles sarcoidosis and has been labeled as sarcoidosis. And so it may be that we're going to come back to interferon as a very important target as we look at the next wave of biologic therapies. And here are some data just to support that point. These are some data from a microarray analysis of sarcoid tissue that Elliot Krauser and we did, looking at mediastinal and lung pathology. And what he did is he looked at networks of related genes, and the network that is most predominant in sarcoidosis is centered around STAT1. That's the most overexpressed network. And in fact, we did some phospho-STAT1 staining, and in the granuloma, it's quite high. And there are other data that have been reported as well that suggest that interferon gamma and STAT1 signaling has a very large centrality to the pathogenesis of sarcoidosis. As you know, it's very hard to make a granuloma without active interferon. If you look at transcriptional regulatory analysis of that same data set, you'll see similar things. Again, STAT1 is in the middle, and some of these other players you've heard discussed during this conference. We won't go through those, but this looks like it may be a very nice target, and there are agents now that can be investigated that might be useful for the therapy of sarcoidosis, and also may be related to this phenomenon of paradoxical granulomatosis during anti-TNF therapy. I think there's time for a few questions, so thank you for your attention.